Yes, hello everybody. Hope you have an amazing day. Today we're going to talk about the biliary system and especially about the gallbladder. The biliary system is the system of of canals on which bile from a liver flows to the gallbladder and then to the duodenum. The biliary tracts include intrahepatic and extrahepatic passages. The intrahepatic passages consist of intralabular and interlabular. The intralabular are the bile canaliculi and the bile ductules, while the interlabular are situated in the interlabular connective tissue in the bile ducts. The extrahepatic passages consist of right and left hepatic ducts, hepatic duct, and common bile duct. The bile canaliculi are thin tubes that collect bile secreted by hepatocytes, and the bile canaliculi are formed by grooves on the lateral faces of hepatocytes. The bile canaliculi form a complex anastomosing network progressing along the course of the liver lobule and terminating in the region of the portal spaces. The bile flows from center of the classical lobule to its periphery, and at the periphery, bile enters the intrahepatic bile ductules or Hering's canals. After a short distance, the ductules cross the limiting hepatocytes of the lobule and end in the interlabular bile ducts in the portal triads. They gradually enlarge and fuse, forming right and left hepatic ducts, which leaves the liver. Intrahepatic bile ductules are lined by cuboidal epithelium, and all other parts of the biliary system are lined by a tall columnar epithelium. Here in this slide, I think it's very clear. As you can see here, this is the cuboidal epithelium, and this is the tall columnar epithelium. Let's just jump to the gallbladder. The gallbladder is the pear-shaped sac that stores the concentrated bile, which is approximately 30 to 70 milliliter. The wall of the gallbladder consists of 3 main layers: the mucosa, fibromuscular layer, and the adventitia or serous layer. The mucosa has numerous branching and anastomosing folds and consists of epithelium and lamina propria. The epithelium is a simple columnar specialized for absorption with an apical brownish border of microvilli, as you can see here in this slide. The gallbladder epithelium includes only the single cell type. It has no goblet cells, as in other structures of other organs. The lamina propria is formed by a loose connective tissue. As you can see, numerous loose connective tissues over here, all over the gallbladder mucosal layer. Also, there is the fibromuscular layer that contains a smooth muscle tissue and loose connective tissue. Here is very obvious. You can see in front of us here in this slide. And the adventitia, which covers the upper surface of the body and the neck. The fundus and lower surface of the body have a serous layer. I think here in this slide is very clear, as you can see here, because this slide contains some of liver and the bile surface. The liver here, this slide is very kind of powerful, because we can see all parts of the liver, starting from the hepatocytes, forming the bile canaliculi to the interlabular and interlabular ducts to the main biliary system. So the mucus forms the deep diverticula that called the Rokitansky-Shov sinus. Let's search for Rokitansky-Shov sinus in this slide. No, this is not obvious. Let's search for one. Oh yes, in this one, this is very obvious. Rokitansky-Shov sinus. This is a place where microorganisms such as bacteria that may accumulate in these sinuses, causing chronic inflammation. So yes, in this kind of places, Rokitansky-Shov sinus, you always need to remember this in cases of chronic inflammation of gallbladder or any of the biliary system. Yes, and that is all for today, and don't forget to like, subscribe, and share all the videos as always, and see you in the next video.